What's up, guys? Today we're going to be learning about protein synthesis. I'm Brooke Fitzpatrick and I'll be the reporter today.
My name's Ashlyn Wilder and I'm going to explain protein synthesis.
And I'm Katyala Shishkina and I'm going to also be explaining protein synthesis.
Ashlyn, what does protein synthesis mean?
So it's your body's way of just making protein. So it's split into two different parts, transcription and then translation.
All right, Ashlyn, can you explain the base pair differences between DNA and RNA?
So in DNA, A pairs with T, but in RNA, A pairs with U. So DNA, RNA.
All right, now what are the building blocks of protein?
Those are amino acids.
All right, now can you remind me what the two steps of protein synthesis are?
Transcription and then translation.
Now, Katya, can you show and tell me where and how transcription takes place?
So transcription takes place in the nucleus. And so first, this DNA strand is unzipped. And we use 1/2 of the DNA as a template for the mRNA to start building. So it starts building using RNA polymerase II. And this is already pre-built, but in reality it would go one by one to match the other base using the RNA rules. And so after it's transcribed, this mRNA strand leaves out and it starts traveling, and in reality it would be encased and capped so that it'd be protected, and it starts to go try and find a ribosome, and that's the end of transcription. Last thing.
Yes, you may.
Okay, and this mRNA strand, typically it's built by using an AUG, initial sequence starter. So our model doesn't have that for convenience sake, but it typically starts with the AUG and then keeps going until it reaches a stop sequence initiator. These would go back together as well.
All right, now which information carrying molecule takes place in transcription?
The carrying molecule made is this mRNA strand.
All right, now can you show and tell me where and how translation takes place?
So as it's traveling through the cell, it finds a ribosome and it attaches to it. And this ribosome reads these mRNA codons, which is these 3 over here, and it brings over these tRNAs, which are carrying an amino acid. And these tRNAs are trying, but through trial and error, to find their correct match based on these anticodons, and there will be a match here. Okay, so now these are connected here. So they connect, pretend these are connected. And once they connect, they find their correct pairing. These disconnect over here, these amino acids, and they're in the correct place now. And now, because they're in the correct place, they create a peptide bond here between each other. And now we have our completed protein. So now what do we do with all these empty ones? Well, they are like the transport buses of our cells, so they just leave and find another amino acid to connect to.
Okay, now which one was the bond formed between the adjacent amino acids?
This is the peptide bond between these.
Perfect, and then is that the completed protein?
Yes, this is the completed protein.
All right, guys, signing off. We were working hard in the hallway. Thank you guys for explaining protein synthesis.